Hello, everyone. Good morning, good afternoon, or good evening. Welcome to the 8th week in the Long-Term Animal Research Seminar Series. We continue this week with Dr. Janet Mann. And before I introduce Janet, I'll make a couple of announcements. First, if you're participating via Zoom, you should see a Q&A tab on either the top or the bottom of your screen. And if you open that tab, you'll be able to type any questions that you have, as well as see and upvote other people's questions. And at the end of the talk, we'll go through those questions, starting with the ones with the most votes. However, if you have a clarifying question that you feel like needs to be addressed during the talk in order for you to understand something, you can type clarification in capital letters at the start of your question. And Janet has said that she'll do her best to answer those clarification questions in real time. Second, recordings of all the talks are available on YouTube shortly after they conclude. So if you need to leave early or know others who are unable to attend live, the talk will be available for viewing after it's complete. Now, I'm very pleased to introduce today's speaker, Dr. Janet Mann, Professor of Biology and Psychology at Georgetown University. Janet received her PhD from the University of Michigan before becoming an assistant professor at Georgetown the next year. And Janet leads 2 long-term studies of dolphins, the Shark Bay Research Project in Western Australia, and the more recently established Potomac Chesapeake Dolphin Project in the mid-Atlantic US. Her lab's research is multifaceted and has made fundamental contributions to our understanding of cetacean biology, including insights into dolphin social behavior, group dynamics, tool use and cultural transmission, and conservation efforts. Every time I read or hear about the work done in Janet's lab, I'm so amazed at the insights that she's able to gain at both the individual and population level in a species that spends so much of its time outside of the view of the people researching them. And I think understanding a species where that's the case requires a different set of logical and analytical skills and obviously different logistical skills than the ones that are intuitive to me. So I'm really excited to hear about Janet's work, and I'll welcome her and turn things over to her now.
Thank you, Matthew. And I'm really pleased to be doing this series with so many esteemed presenters who are doing longitudinal research. And I'm looking forward to hearing your questions and comments on the work. So I've listed the names of a number of my collaborators, a lot of my grad students, and whose work I'm going also to highlight today. And I want to point out my colleague, Celine Frere at University of Sunshine Coast, who does the genetics work with us. So I will get started, and first I want to credit Jean Altman. I actually started as an undergraduate. I was at Brown University, but worked with Jean Altman at the Amboseli Baboon Project back in the 1980s, so a long time ago. And I also want to point out, after a year I spent in Amboseli, that my favorite t-shirt, you might not be able to see that well, but I actually have a whale on my t-shirt. And what I learned from Jean was how to establish a long-term study. And so in graduate school, I was always interested in all animals. I was sort of more on the primatology side of things. But my advisor in graduate school brought me out to Shark Bay when she wanted to initiate this long-term, a study of mothers and calves. And then when she decided to return to dog work that she got interested in, I kept going with the Shark Bay research. So here I am at the bow of the boat. So much of the Shark Bay dolphin research is modeled on essentially Jean Altman's Amboseli project. So to take you to Shark Bay, it's 13,000 miles away and in Western Australia. And our field site is in the Eastern Gulf, and this is roughly the area, about 300 square kilometers. So some of the things that are fascinating about dolphins are listed here, and these are the things that we're broadly interested in and that I'm going to talk about today. And the thing I'm going to tell this story through is through their life histories. So everything is going to be told sort of through the life of male and female dolphins and how all of these other factors come into play. And fundamental question is, what are the significant life challenges and trade-offs during development? And to do this, I'm going to focus on several time periods. First, the young of the year, or the first year of life. Then the calf period, which is up to 4 years of age, which is the average weaning time. So that's when they stop nursing. The juvenile period, 4 to 10 years of age. And the young adult period, which is 10 to 25. And then the older adult period, 25 to 40, although I'll talk about the adult period generally. And I'm also going to be focusing on females much more than males, but I'll tell you, the males come in certainly in the early period, and then when I return back to adulthood. So Dolphin Society, just a bit of background, is a very dynamic fission fusion society, meaning that they are not in a stable pod. The average group size is 4 to 5 dolphins, but they can be alone all day or with 30 or more individuals. And that changes multiple times an hour throughout the day. Males, as many of you probably aware, form these multi-level alliances, sort of represented there below, and it's all about getting access to females. So the pairs and triplets of sort of first order alliances collaborate with sometimes their second order alliance to work together to gain access and sequester cycling females. Females have a looser kin-based network structure, which I'm going to talk about today. And females first calve at about 11. The first calf is 11 years of age. The average first calf is at 13. And their maximum lifespan is into late 40s, possibly early 50s. In terms of the life history trace, this is a little bit of the methods, but I'm going to not focus that much on the methods today. We do know the birth years for over 800 calves, and the older individuals, age estimates we can do by size and ventral speckling, but we know the birth years of calves born since at least the 1980s. We have the calving success or reproductive success data for 257 females, which I'm going to show you. And weaning is determined by cessation of infant position, which is shown in the background here. When the calf swims under the mother, that's when they keep nursing. But also their association drops well below 50% post-weaning. Death dates are determined by their disappearance for calves under 3 years of age if they're not with their mother anymore, and then we never see them again. And both sexes are philopatric, so if we don't see them for a number of years, and since nobody leaves the area, we assume that they're gone. Behavior, we have 37 years of associations or survey data, sort of snapshot of groups when we go and survey out in the water. And then we do detailed focal individual follows, which I'll talk about all of these. I'm kind of not going to differentiate in the talk which data I'm necessarily using, but happy to answer your questions about methods. Molly McKenty, who made an appearance at Joan Silk's talk, because she also did baboon work in addition to hyena work and whale work and some other things, has been analyzing our long-term, our mortality data, and for about 700 dolphins. And here, this is just for the up to age 10. The highest mortality is in the first year of life at 23%. And by age 3, 33%, so another 10%. And that's because that's the minimum weaning age, even though the average age is 4. And 41% are gone by age 10. So that gives you something of the challenges for calves and juveniles in terms of mortality. And to describe a little bit of the first year, so I'm going to go through kind of these different life histories. So now we're in the yoi stage, or young of the year, and the challenges for both sexes. I mean, for a mammal that needs to live in the water is, of course, breathing, and that's even challenging for a newborn calf because their breathing is conscious, and they have to stay with the mother, so they're precocious and have to swim and be able to move quickly right from birth, avoiding shark predation, coordinating with the mother, particularly diving. She usually can't feed very often in the early weeks after she has a calf because she can't really dive. And the calves have to learn in that first year also how to join and leave from the mother, much like the fission-fusion dynamics. They sort of emulate that pattern early on, joining and leaving from the mother, going on separations. In the first year, they also learn to catch fish, and they have to learn the network of multiple individuals coming in and out. Their social behavior is highest in the first year of life for the calves because they do play a lot and interact a lot. And their foraging is lowest in that first year. Before they're weaned, they also have to develop specific foraging skills, and I'm going to talk about those because they have extremely diverse foraging tactics, which they learn primarily from their mother, and their female bias, these foraging tactics, and that females do many of the diverse tactics. They coordinate heavily with their mother, of course, and have to learn to kind of separate and rejoin. For males, the challenge is to develop male-male bonds, particularly, although females develop bonds within their mother's network. And I'm going to talk a little more about this. And again, the fission-fusion dynamics of separating and rejoining with their mothers is an important feature of their pre-weaning. And that also allows us to look at some of these emerging sex differences. And Quincy Gibson, who's now at University of Florida, did work on some of the sex differences early pre-weaning. And just to explain this is showing you the when calves are separated from their mothers on the y-axis, and when they're with their mothers on the x-axis, and their time in large groups, basically when they're with their mother and away from their mother. And females, the solid line, basically do what their mother does. If their mother's fairly solitary, when they separate from their mother, they're also solitary. And if their mother's very sociable, when they separate, they are gregarious similarly. For males, it's actually the opposite, which is quite interesting. When the mother is not very sociable, then the calf makes an effort to be in large groups when on separations. And the more sociable the mother is down here, the less sociable then it becomes on separation. So basically, the males have to work harder. They have to be they're more gregarious on separations when the mother is less sociable, suggesting that they have to go sort of to greater effort. And who do they associate with? Well, Maggie Stanton, another one of my former graduate students, who's at University of New England now. The males seek out other male calves. So they're already at a young age, and this is just shown in that first more than expected, basically this is showing you the positive values of more than expected. A male calf is seeking out other male calves. And calves of both sexes avoid adult males. That's just what's showing down here. They associate much less than expected, especially when they're away from their mothers. So this is basically male calves are very attracted to each other from a young age, and the daughters are basically doing what their mother does. But also mothers do something for the daughters, and Madison, another one of my former graduate students, published a paper on the diving behavior. And as I've mentioned, foraging is pretty important for the females in particular. And our data suggests that the mothers adjust their diving behavior for their female calves, and that's shown at this young ages here. Mothers are doing shorter dives. This is the mother's dive durations on the y-axis, and they actually shorten their dives for their daughters, and they don't do any of that for their sons, which is shown on the right. Basically, for their sons, they just forage as they like, and that's when they're close to when they're with their in contact. So when the calf is basically in baby position or very close to the mother, this is when the mothers are adjusting their diving behavior. So this enables the mothers to be able to stay with reverse that, it enables the daughters to stay with their mothers while they're foraging, so they can observe their mothers, I think, much more closely. And so it suggests that the mothers are enhancing their daughters' foraging skill and not making as much of an effort. And this is also pays off in the sense that female calf diving competence actually improves much faster than the male diving competence. So already you're seeing a difference, females with foraging and males with social behavior. And that's going to be a running theme. The activity budgets also reflect that. Eva Krischik, another former graduate student, published research on the transition, the 2 years like pre-weaning and then 2 years post-weaning. And the females increase a lot in foraging. There isn't a big change in the males' activity budgets from pre to post-weaning, but the daughters, just in the juvenile period, it's all about food. And I'm going to go back to mortality and the sex differences in mortality, and then talk a little bit more about the juvenile period. So in terms of calf mortality, we don't find a sex difference. And it's a little confusing because you see this big green line below. So here the blue line for males and red for females. No difference in sex difference in mortality. These are the unknown sex. So it's basically, if they die in the first year, we don't get a chance to sex them, and that's all that means. But we still don't think that there's necessarily a sex difference in mortality pre-weaning if males died early and we just didn't know their sex, because the adult sex ratio is 50-50. So you would need more male births for that to happen. But post-weaning, and when the juvenile period starts, is where you see higher male mortality. So juvenile male mortality then becomes higher than female mortality post-weaning. And this seems to be linked also to the social dynamics. So the daughters, as I said, are sort of more linked to the mother and the mother's network, whereas the males kind of have to work harder to eventually establish an alliance, but are attracted to other young males from an early age. But the mother has an effect on that. And I should mention, so it's a 12.8% mortality rate in the juvenile period. And that's mostly males. Very few females die in the juvenile period. And going back to Maggie Stanton's work, she looked at how eigenvector centrality is a measure of like how many important sort of individuals you're connected to in your network as a calf predicted survival for males, but not for females. So this top line is showing you the females, and it wasn't related to the eigenvector centrality, but for males, there was a strong relationship. So if they had high eigenvector centrality, essentially while they were with their mother, that predicted their survival. And most of that was in the couple of years post-weaning. So having early social connections seemed very important for male survival. So again, back to the social dynamics. So these relationships that they have to work hard to develop, whereas the females, it doesn't seem essentially to matter as much because they have their mother. The challenges for juveniles, now I'm going to some work Ali Galiza did as she was with me as an undergraduate. Many of you know her now at Duke University, as she is working on the Amboseli project, and so doing the reverse of what I did, I guess. And she looked at sex segregation and their behavior during the juvenile period, and looked at all juvenile groups. And it shows how much the males have to sort of develop their own network, whereas the females can sort of keep their mother's network, which is mostly other females. And this is just showing you the association strength. Females are already, they're already sex segregated in that females associate more with females. And this is especially true for males during the juvenile period, so much higher than expected. Both sexes, again, are still avoiding adult males, but the sex segregation really is accentuated during the juvenile period. And more on this topic, again, the finding, repeating what Ev found, the foraging activity is much higher for females in the juvenile period, much higher than males. That's what's being shown in this first figure. And then in the survey data, the social behavior is much higher for males, both in mixed groups, but also all male groups. So for females, it's all about the foraging in the juvenile period, and for males, it's all about social skills through male interactions and developing these bonds. And I do want to point out Taylor Evans, who's a graduate student currently in my lab, and she's looking at the social phenotypes and the stability of these phenotypes. And we find very high stability from calf to adulthood. So this is showing you the repeatability from when, for males and females, females square and males are triangles. The red is sort of the expected, and the upper black bars show you the repeatability. As you can see, time alone with either when they're young, it's alone with your mother, but when you're a juvenile and adult, that has very high repeatability, less so as you might expect for males because they do have to be more social, but very, very strong for females. And in large groups, you also see this very high repeatability, high gregarious. The small groups, which are 2 to 6 animals, is not that repeatable, and that's because that's kind of the typical group size. It seems like everybody does that. And then similarly with degree or the number of associates, there's very high repeatability for both sexes, and for females for foraging, as you can see up here. Social behavior varies because that, as I mentioned, in the first year of life, calves socialize a lot. So as calves and juveniles, they socialize a lot, and social behavior declines strongly with age, so it's not very repeatable. To give you a sense of the network dynamics, I'm going to play you a little, a short video. This is a male on the left, Cookie. He's in the middle of the blue here, and that's his mother, Crooked Fin. And the thicker lines are the strength of the interactions. I'm just showing you the top 15 associates, otherwise it'd be too messy. And it's going to play the calf period first, then the juvenile, it'll slow down, and then there'll be the juvenile period, and then you'll see the adult network. Similarly, this is a female calf over here, Peglet, and her mother Square, who died in her adulthood, so you'll see Square will disappear, but her sister Squarelet, and then a younger sister, Rhombus, will show up in the image. And you'll see, what you'll see happen here is the blue male will become more blue as he develops his alliance partners. And so you'll see some of these same guys, Smokey here and Jesse, and will strengthen bonds, and others will come in. And the female network, it'll sort of become a little more loose, you'll see how it's similarly structured to how it was when she was a calf. So I'll just go ahead and play it and talk you through. This is the calf period. And then you see the juvenile period, and some of the males are coming in. Smokey comes in. This is still in the juvenile period, and then we're getting to the adult period, and you could see the strength of Cookie's bonds with the males now. over time are like very strong, and it ends with this very tight alliance and this other male up here who happens to be his father, whose alliance partners died, who joined up. So you can see how that looks very different from what he had as a calf, and but he still has some female connections there from the beginning. For the daughter, for Peglet, you can see she's still strongly connected to her female kin. And then her mother is still there, but she's still with her other female kin. Her mother's gone now, and she has this kind of loose network, and her strongest bond is with actually her younger sister, who's up here. So that gives you a sense of what the network dynamics then look like into adulthood. So you can look at a sort of a male network and a female network. The costs of reproduction for females are very strong, and this is something that Molly McKenty is looking at, and for her PhD. And to give you an example, remember the males had higher mortality before age 10, and now it switches. And in the early years of reproduction, female mortality is now higher. And we think that this is because for several things. One is just the tremendous energetic cost of calf production. But it also, we think it's linked to male harassment, these alliances, and their sequestering of females, and that this exerts costs on females. Their lifespans are not different. This is another interesting feature, is that usually males have shorter lifespans than females, and I'll return to this at the end. So right now, there just doesn't seem to be a sex difference in expected lifespan. And so I'm going to talk a bit about adulthood now. And I can't talk about female adulthood without mentioning the diversity of their foraging tactics. And here's just a few. And we've documented close to 20 different foraging tactics. Some are rare and some are much more common. But this trevally hunting, which is done by two families, snacking, which is widespread, and that's a belly up foraging. Trevally is just giant fish that they catch. It's very difficult. The snacking, belly up foraging. Another very difficult technique is beaching, which they do, which is two families three families actually that do it in different locations with slightly different techniques. Shag robbing, cormorants are called shags in Australia. So they do steal fish sometimes back from the shags, but there are individuals who just hit the shags when they catch a fish and steal their fish. Sponging, which is the most famous foraging technique, where they use conical marine sponges. I'm going to talk a little bit about that today, but not much. And that's a select group of females who basically do a lot of that behavior. They use the sponge to protect their beak and to find prey that are well hidden in the substrate. And then shelling, which you might have heard about, which is a technique where they grab trumpet shells, which is a giant snail, or baler shells that are empty at a fish that is presumably swam into, and then they hold it up out of the water to drain the water out and get the fish. And so its females are biased on all these behaviors. There are no male beachers. There are fewer male spongers. There are no fewer trevally hunters, etc. And it takes a lot of technique to develop these tactics. Eric Patterson, one of my former grad students, focused on this and looked at the tool use. And I think one of the more interesting findings was that it took many years to get good at tool use, and that meaning that they were more efficient with their tools. So basically they change sponges, the sponge dies, they take it off the bottom, and then it's good for sometimes an hour or more. But the young ones just drop their sponges right away, either because they don't select the best sponges, And it's the older individuals who can keep their tool for a long period of time. And there's other things like they find females, older females are better at finding sponges more quickly, and a number of things. And maybe the youth are just pretty wasteful with their with their tools. And all of this heterogeneity in female foraging behavior and their social behavior as well, which I'll talk about a bit, it means that there seems to be a lot of heterogeneity also in their calving success. So more variance than you would expect. And I just don't know if any of the other long-term researchers out there can tell me if they have anything like this, because we've been looking through the literature. We can't find another example that's quite like this. So about 20%, 21% of the females have had no calving success. And then it's sort of normally distributed after that. So this is something that Molly is focusing on trying to figure out why we have such a high number of failed females. I should note that if they fail early in life, and then they just never are successful. So it's not that females suddenly have their first calf and are successful at age 30. They have to succeed by their early 20s. The average for all of them is 0.099, or put another way, a female successfully calves on average, and meaning a calf survives to age 3 every 10 years. So up here, the super successful females. Celine Frere, who I've collaborated for a long time, looked at how this relates to both social and genetic effects, and found basically a stronger social effect for females on calving success than genetic effect. Here's one way of showing what is a fairly complicated result, but basically females' preferred associates, which are on the x-axis, this is the mean preferred associates, and their calving success on the y-axis, and the solid line is females of low relatedness, and there was a stronger relationship of spending time with those of similar calving success if you were less related than if you were more closely related. So this could mean a number of things. One is just that females with calves tend to hang out together, and those are going to be the successful females, or success may help beget success because of those relationships. But this is something we're trying to still better disentangle, but there is a social effect, but we don't fully understand what that social effect is. And many of you know Vivian Ferrograd, who worked with me as an undergraduate and then went to Duke University and did her PhD, still in the Shark Bay work, and is now postdocing with me. And she is focused very much on social and genetic relationships between individuals, and has shown in some very elegant work where you can control for the overlap, the spatial overlap between individuals, because in this dynamic vision fusion where you've got animals moving around all the time, you need to sort of control for that. But basically that matrilineal bonds are strong, as we knew, but not controlling for their overlap. So they do preferentially affiliate with female kin, but also with non-matrilineal kin. So there may be some other kin recognition mechanisms that need to be explored, because males have nothing to do with females raising their offspring. It's just a mating and then they're done. And females also, interestingly, form a number of bonds with non-kin, and trying to understand those relationships. So females do form social bonds outside of their kin groups, and they do that regardless of how many kin they have available. but matrilineal kinship is the substrate of the society. I should say also from other work, the alliance members and the males are not related. They tend to be the same age cohort. So they tend not to be formed, unlike with lions and others, they do not form alliances with kin. Since menopause has been shown in other toothed whales, notably killer whales, but also pilot whales, short-fin pilot whales, and false killer whales, and possibly belugas and narwhals. But these bottlenose dolphins do not show menopause. They do have strong female reproductive senescence, as I mentioned. This is Caitlin Karniski's work. And females would be much more successful with calving at an early stage, and then they slow down. They have both higher mortality and as they higher calf mortality as they age. But they also have terminal investment. So if it's their last calf, they tend to wean them much later. And this protects them from basically investing in another calf and a big energetic investment late in life, which they might not survive, but also ensures the survival of their last calf. And so the one, in fact, shown in the background here, nursed for 8.5 years. So the last born nurses for a very long time, but there's no difference in the firstborn and the middleborn. And unlike in primates and some others, where it seems like maternal care is challenging, we don't get a firstborn mortality effect at all in the population. So the final piece, I'm going to return to social bonds and survival for both sexes. And here you can see the whole sort of sharp bay network for individuals that we know fairly well. Red is the females and blue is the males. And this is a cumulative snapshot. And dolphins vary widely in their gregariousness and their bonds. So the males are kind of more similar to each other because they kind of have to be in an alliance. So they have to have a buddy, at least one or two, or sometimes more than 10. But they have to be in an alliance of some sort in order to have any hope of surviving and getting access to mates, access to females. For females, they can be very solitary, pretty much like the spongers are, and some of the other females are, or they can be extremely gregarious. And so this is puzzling because people usually think, oh, more social is better, and having more buddies or more friends is better. And so this made us think that maybe if individually differentiated social bonds are really important, but they vary widely between individuals, then it might be the change in those bonds, which has survival consequences, not the absolute number of bonds or not the strength of those bonds. And so it's not being highly sociable or highly solitary. It's basically when you lose or some difference in those relationships. And Rob Rankin, who postdoced with me for a year, looked at this using sort of the long-term network dynamics, so leveraging our 30 years of data, looking from using 2-year intervals where we would have the sort of network taking those chunks across many years, and to look at whether we can predict whether a dolphin lives or dies based on the change in their network. And we used both kind of a predictivist model and inferential, and they came up with identical results. And a couple of interesting things. We threw in a lot of different variables for males and females. And what came up really strongly was the closeness centrality, or a form of the social position. And so it was the drop in closeness centrality, regardless of whether you were pretty sociable or solitary. So it was the loss of your social position. And that could be through other individuals dying or just being booted from your network, basically. So that had an impact, that predicted death, and even stronger effect than age itself. Like age is a very strong predictor of death, obviously. But so this was really interesting. So it shows for both males and females, this is important. It came out no matter how you looked at the data. Another interesting effect that we want to look at was what's called the widowhood effect, and we expected that this might come up for males, and it did. And that's, as some of you may know, for humans, when one partner dies, the other is more likely to die within a year. It's especially true that husbands are more likely to die when after their wife dies, and less so on the reverse. So after a husband dies, wives are more likely to outlive them for longer. But we did find this widowhood effect for males, sort of with their, basically with their closest associate, which is their alliance partner. And in this complicated figure, this is showing you the log odds of dying. And basically all of these orange stars you see shows you the widowhood effect metric, meaning that the odds of dying for a male went up when he lost his closest associate, and the effect was just not really there for females. So this suggests a strong social buffering effect. It's obviously stronger for males than for females, but that doesn't mean that the social bonds among females are not important, because as you've seen today, it has an impact on their calving success as well. And so to summarize some of these issues, the females, it's all about the foraging and a benefit of the maternal network. I'm also showing you here aren't the Shark Bay dolphins, but actually the Potomac Chesapeake dolphins, to give a little shout out to Ani Jacoby, who has been doing that work from Duke, and some of you know her. But for the females, it's all about foraging, and they benefit from kind of the maternal network, as do the males. But for females, it's more of a lifetime kin group and other relationships that they form. They have very high behavioral heterogeneity and variation in reproductive success. And we think the costs of reproduction are very high for females, and male harassment probably contributes to that. For males, it's all about social competition, even from a very early age. And that shows up in their vulnerability early on. But if they're successful and they have a partner, that they get perhaps a social buffering in adulthood. And that's shown with the widowhood effect. And but for both sexes, the loss of social position predicts survival or predicts mortality for both sexes. So indeed, their social lives are very important. And I'm going to finish with two things. First, citing the many different supports that we've had over the years. And then while I take questions, I'm going to show some images of Shark Bay that were professionally shot, not by me, just so you could see what it looks like in motion. And thank you for your attention, and I hope everyone's still awake. And I'm happy to take questions.
Well, thank you very much, Janet. I have about 1 million questions, but we'll start with the questions from the audience. Let's start with Mark Hauber, who has a kind of logistical question. He says, regarding the unsexed calves dying early on, is there a chance to get DNA samples from the calves before they perish for parentage and sex tests? And maybe you could talk a little bit, if you do get biological samples, what you are able to use those for.
Yeah, the problem is that we use a biopsy. These are animals moving fast in the water. We don't tag them or capture them. So we use a biopsy gun, and we we don't shoot the calves because until we wait until they're at least 2 years of age, when they're like pretty big. And so we haven't had any mortality or problems from like the actual biopsy, but I think we'd just be too nervous. You can get DNA from poo if you can find it. And like with dolphins, it's sort of just really, really hard. We occasionally have captured poo from adults, but from infants, like never. So that is a problem. And we never get carcasses because it's Shark Bay and it's full of sharks. And so there's a little baby and we just can't get it. I'd love to. So you could see, we could tell when they're pregnant because they're very blubbery. Or soon right after birth. There are some of these females we've seen clearly visibly pregnant, and then we never see them with a calf. So we don't really know. We think that one of the problems is for females that lose their calves when they're young, that then they start cycling out of season. They do have seasonal births, and that might put their calves more at risk, and that the females who are cycling all the time, essentially because they keep losing calves, that they experience more male harassment, and it's a reciprocal effect that contributes to their failure.
And I think Ashley also has a follow-up. For those females whose calves that don't survive, how quickly do they return to cycling? Is that pretty immediate?
I'm gonna stop sharing. Sorry, so repeat that again.
So for the females whose calves don't survive, how quickly do they return to cycling and start mating again and become pregnant?
Yeah, so we actually have somebody looking at that right now. So they can return if they lose the calf when it's very small, like only like less, just within a couple months after birth. They can become pregnant within as quickly as a month. But if it's longer than that, it usually takes them, 6 months they'll usually wait till the next breeding season, essentially, most of the time. And if they lose a calf who's even gone further, like a year old, it might take them even longer because they're depleted, and so they have to recover before they start cycling again. But we are looking at that.
Anna Hewitt asks, do males with less sociable mothers have a higher fitness? So do their offspring survive longer or more often?
That's a good question. There is definitely a skew in male reproduction. So we have a number of very successful males. Unfortunately, we don't know their mothers because these are males who are they start being successful at best late teens and then mostly into their 20s and 30s. So the ones that we have genetic data for, we don't know their maternal history so well, but I suspect yes, based on the little bit that we know. So yeah, I suspect that might be the case.
And then Susan Alberts asks about the widowhood effect. She asks, are the closest associates of males always males, or are they sometimes females? So when you refer to the widowhood effect, is that the loss of same-sex social partners or opposite sex?
Yes, it's always the same sex. I mean, there some of the old ladies and old men kind of the males hang out together a little bit, but it really is same sex.
And I had a question about the apparent lack of difference in lifespan between males and females, which, as you noted, is so different from primates. And it sounded like you think that that's because of relatively high mortality for females, right, due to male harassment and reproduction. Is there something that males might do? I'm just curious what the differences in societies are here that might account for that lack of difference. Are males protected in some way from costs of male aggression? Are females more vulnerable?
Well, we think males are protected from social buffering. Basically, they're, there's a trade-off for them. They have to share. I mean, they have to share in the matings by being in an alliance, but they benefit by having a social partner that buffers them. And that's why we also think we see the widowhood effect for males. So that's rare in the animal kingdom, having those long-term alliances, and but that might benefit them in terms of them being able to stick around for a long time.
And I had a question. You also suggested, it sounded like you thought that reproduction potentially is costly to females in and of itself.
Mm-hmm.
I wonder if you've looked at parity as a predictor of female death, such that females who are actually having more kids after controlling for chronological age, maybe they're too tightly linked, but
Mm-hmm.
after controlling for chronological age, if more parous females are more likely to die.
So far, it looks like the opposite.
Okay, huh?
So it could be. So one of the things we're trying to tease apart is like, are these, females who have, I should say not parous, but those who have successfully calved. So it's those low success females that have a higher mortality. And Molly has presented these data. So like the low success females are much more likely to, the zero success females, the totally unsuccessful females die young. So we don't know if it's a condition thing or there's just some other thing that's gone wrong. And that's something we're trying to figure out. So, but success, because of reproductive senescence, the female's calving success rate goes down with age. So the longer they live, so we have females who, have lived well into their 40s, but the longer they live, their kind of calving success rate is going to go down because of reproductive senescence, like overall. But it seems like those are, successful females live a long time.
And then I had one more question, which is about male harassment at different cycling stages, different reproductive stages. So males harass cycling females. Do they harass pregnant females and lactating females? And how much of a risk do they pose to fetuses and infants? Or calves, rather.
Yeah, there's two parts of that. So I should have shown some of Meg Wallen's work. She was another one of my grad students she looked at this. The adult males really know what they're doing in the sense of we don't know how they know when females are cycling, but they act like they know exactly when females are fertile because they, when you look at who they're associating with and how long they stay with them. So however they do it, and whether they do it by sonar, and they can tell, because they have very sophisticated sonar, can tell like when the follicle, actually see the follicle and know the female's reproductive state, or whether it's chemical cues. They seem to know when females are cycling. Females, and when they have a calf, males and females avoid each other, but we don't have any examples of infanticide. I only have one really aggressive incident where this was with an older calf, and I think it was more about the particular male calf and his brothers than about infanticide. So we don't have any records of infanticide, although infanticide has been documented at other sites for bottlenose dolphins. And we think that the lack of infanticide might be linked to things like the paternities are like all local males, like they know the males they're mating with. They're not really encountering strange males. So these are familiar males, and males just aren't gonna, they're not gonna kill their own, but they might kind of, in essence, like just not be, it's not a good strategy for them most of the time. And the chance, even if they killed the baby, the chance that they'd be around when the female was cycling again is pretty low. So even if I know I'm not the daddy, I never made it with this female, they would have such a small chance of being with that female, you know, a month or 2 or 6 months or a year later when she was cycling again.
Well, I think that's all the questions we've got. Thanks for such a robust and fascinating talk, and thanks for giving it. It's great.
Thank you.
And to everyone out there, next week, we're going to have a talk from Alistair Wilson at Exeter. So join us at the same time, same place next